this topic, we will learn more about the innate defenses that are crucial to preventing and resisting infection by forming our first and second lines of defense. As you will recall, the first line of defense, the surface barriers, are like the walls and moats of a medieval castle. They are designed to keep invaders out and include the skin and mucous membranes and the chemicals they secrete. The second line of defense, the innate internal defenses, responds to pathogens that have entered the body as soon as they are encountered. In our castle analogy, innate internal defenses corresponded to the castle guards. In the body, these include cells and chemical substances in the extracellular fluid. Many potential pathogens are either prevented from invading the body or are quickly eliminated following invasion. When infection does occur, innate and adaptive defense mechanisms cooperate to clear pathogens from the body. Your goals for learning are to describe the physical and chemical surface barriers to infection, to describe the cells, antimicrobial proteins, and processes that form the innate internal defenses, to understand the purpose, sequence of events, and cardinal signs of inflammation, to recognize that innate defense mechanisms may be insufficient to prevent or clear infections, to learn that innate and adaptive defenses interact in many ways, and that innate defenses often trigger and define the nature of adaptive defense responses. Here's what you need to know. What enzymes are and how they work. Lysosomes contain hydrolytic enzymes. The nature of proteins, nucleic acids, and polysaccharides. What endothelial cells are, and that capillary walls consist only of endothelial cells and a basement membrane. To see definitions of terms, click the bold red words. Even though our environment is teeming with potential pathogens, most of them never cause us any problems. This is because our first line of defense, which includes physical barriers such as intact skin, and the mucous membranes of the respiratory tract, the gastrointestinal tract, and the genitourinary tract, prevents most pathogens from entering the body. Skin acts as a physical barrier to infection. 3 properties of the skin are key to its ability to resist invasion. First, the outermost layer contains the tough protein keratin, forming an abrasion and water-resistant surface. Second, specialized intercellular junctions, such as desmosomes and tight junctions, hold skin cells tightly together. Third, skin secretions are acidic and contain chemicals that make the skin an inhospitable place for pathogens. One of these secretions is the enzyme lysozyme, which can digest the cell walls of certain bacteria. Click the skin to see it resist pathogens. Mucous membranes line all body cavities that are open to the external environment. These include the digestive, respiratory, urinary, and reproductive tracts. Click the mucous membranes. Generally, mucous membrane secretions are acidic and contain lysozyme. In fact, Lysozyme is found in virtually all surface secretions, including sweat, tears, saliva, and milk. In certain locations, mucous membranes have special properties. For example, in the stomach, pathogens that may have entered through the mouth encounter both digestive enzymes and a very low pH. The digestive and respiratory passageways are lined with sticky mucus that traps pathogens. Click the trachea to see mucus trap pathogens. Pathogens and other particles suspended in the air, such as dust and pollen, enter through the nasal cavity. All but the very smallest of these are trapped in the thick mucus layer covering the epithelium of the nasal cavity, pharynx, trachea, and bronchi. Cilia move the mucus down from the nose or up from the bronchi and trachea toward the back of the throat where the mucus is swallowed. Stomach acid and enzymes kill most of the trapped pathogens. Click the cilia to see them in action. The second line of defense comes into play when a pathogen has penetrated the surface barriers and entered the body. Second-line defenses attempt to limit the spread of pathogens through the body by taking a multifaceted approach to pathogen elimination. Although these innate internal defenses are fast-acting, they are nonspecific and provide only a kind of crude protection against any and all pathogens that enter the body. Think of them as being like the guards inside the castle. Click a guard to see the 5 lines of innate internal defense. One line of innate internal defense consists of the phagocytic cells, primarily the neutrophils and macrophages. A second set of cells, the natural killer or NK cells, kill body cells that have turned traitor by becoming either virus-infected or cancerous. Another line of defense does not involve cells at all, but rather antimicrobial proteins, such as complement and interferons. The last 2 lines of defense are neither cells nor proteins, but processes. They are inflammation and fever. Click the phagocyte to continue. Sometimes the innate defenses are no match for invading pathogens, just as a massive invasion can overwhelm the guards on a castle wall. In this case, the armies of the adaptive defenses must be called out. Having already met the enemy, the innate defenses can influence the kind of response the adaptive immune system makes by passing chemical messages to the adaptive immune system, rather like this guard gesturing to his friends. This process is one of the many ways in which the innate and adaptive defenses work together. Recall from the anatomy review topic that neutrophils and macrophages are the 2 main types of phagocytes. These phagocytes, considered part of the innate defenses, perform essential functions for both innate and adaptive immunity. Let's examine these remarkable cells in more detail. Neutrophils are the most abundant of the leukocytes, making up 50 to 70% of the white blood cells. They are normally not found in healthy tissues. Neutrophils are the first cells to leave the blood and enter tissue in response to infection or trauma. Click the neutrophil. Neutrophils are recruited by chemicals released by tissue macrophages and injured tissues. Neutrophils are short-lived cells that can only phagocytize a few pathogens before they die. Click the monocyte. Monocytes follow the influx of neutrophils into the affected tissue. These newly arrived monocytes transform into macrophages, reinforcing the resident macrophages that are already present. Click the monocyte to see it turn into a macrophage. Unlike neutrophils, macrophages are normally present in tissues. They can either be free macrophages, which wander on patrol through tissues like guards, or fixed macrophages, such as Kupffer cells in the liver and microglia in the brain. Fixed macrophages stay within one tissue like a posted sentinel, ready to signal and fight off invasion. Unlike the neutrophils, macrophages have a voracious appetite and can phagocytize many pathogens or dead cells in their lifetimes. Both neutrophils and macrophages engulf pathogens and destroy them inside the phagocyte using lysosomal enzymes and highly reactive chemicals. If the pathogens are too large to engulf, such as worms, phagocytes can also secrete their destructive enzymes onto the surface of these pathogens. Click the macrophage to see a summary of similarities and differences between macrophages and neutrophils. Pathogens that enter the body are often rapidly ingested by phagocytes. This process begins when a phagocyte recognizes and binds a pathogen. You might wonder how the phagocyte knows that something is a pathogen and not part of the body. Phagocytes use special cell membrane receptors, such as the mannose receptor and the toll-like receptors, or TLRs, to recognize and bind molecules that are found only on certain pathogens, particularly bacteria, and not on normal body cells. Click a toll-like receptor to learn more about them. At least 10 different toll-like receptors have been identified on human phagocytes, each binding to a different type of pathogen molecule. When phagocytes recognize a pathogen, 2 events are triggered. The first is the ingestion of the pathogen. The second is the release of chemical alarm signals that mobilize other cells of innate and adaptive immunity. Click the phagocyte to see it bind pathogens. Once the phagocyte detects a pathogen, it engulfs the pathogen by extending its cell membrane and cytoplasm around it, bringing the pathogen inside its cell body wrapped within a vesicle. This vesicle is called a phagosome. Click the phagocyte to engulf the bacteria. The phagosome contains the pathogen and begins the killing process. Because there is such a wide variety of pathogens that phagocytes come up against, they require a correspondingly wide range of techniques for killing. First, hydrogen ions are pumped into the phagosome, making it acidic. This kills some pathogens outright and makes it difficult for any survivors to replicate. Click the phagosome to acidify it. Second, enzymes on the phagosomal membrane begin converting oxygen into toxic reactive oxygen intermediates, such as superoxide and hydrogen peroxide, a process called the respiratory burst. At the same time, enzymes in the cytosol begin producing toxic nitric oxide, which diffuses into the phagosome. Click a bacterium to induce a respiratory burst. Killing is enhanced when the phagosome fuses with a lysosome inside the phagocyte, forming a phagolysosome. The pathogen is exposed to potent hydrolytic enzymes that can digest the pathogen, to antimicrobial proteins called defensins that poke holes in bacterial membranes, and to enzymes that convert reactive oxygen intermediates to chemicals that are similar to household bleach. Drag the lysosome to the phagosome to form a phagolysosome. Pathogens and the immune system are involved in an evolutionary arms race. Many pathogens have evolved strategies to avoid being killed by phagocytes. Try dragging the macrophage to one of the two kinds of bacteria, encapsulated and unencapsulated. Now try the other kind. One strategy certain bacteria have evolved is to enclose themselves in a capsule that makes it more difficult for phagocytes to grab them. In response, the immune system has evolved molecules that can coat these bacteria and provide handholds that allow the phagocytes to bind and engulf these bacteria. This process of coating bacteria to enhance phagocytosis is called opsinization. Two immune molecules that can act as opsinins are antibodies and complement. We will learn more about both of these later, but here we will focus on antibodies. Click a bacterium to coat it with antibody. Phagocytes have receptors that attach to the opsinins. The opsinins form a link binding together the pathogen and the phagocyte, triggering phagocytosis. Drag the opsinized bacterium to the macrophage so that both its receptors attach to the antibody stems. There are several other strategies that pathogens have evolved to escape destruction by phagocytes. These include secreting molecules that block the fusion of lysosomes with the phagosome, developing resistance to the effects of lysosomal enzymes and reactive oxygen intermediates, and finding ways to escape the phagosome, take up residence, and replicate within the cytoplasm of the phagocyte. The bacterium that causes tuberculosis is known for its ability to hide out and replicate inside macrophages. In response to bacterial strategies, such as those already mentioned, the immune system has evolved a counter-strategy. Certain T cells of the more sophisticated adaptive defense mechanisms can enhance the entire killing process within the macrophage. This enhancement only happens when the macrophage presents antigen from such bacteria to the T cell. The interaction between T cells and phagocytes is another example of the interaction between innate and adaptive defense systems. Click the macrophage to see it try to destroy the tuberculosis bacterium. Enhance the macrophage's killing ability by dragging the T cell to it. Natural killer NK cells are unusual. They are a type of lymphocyte, and yet they are involved in innate immunity. They make up 10 to 15% of the lymphocytes circulating in the blood. Let's compare these unusual lymphocytes to B and T cells. Click the NK cell. Like T cells, NK cells kill the body's own cells under two circumstances: if those cells have been invaded by intracellular pathogens, or if they have become cancerous. NK cells also attack transplanted tissues, playing a role in the rejection of transplanted organs. Click the T cell. NK cells are larger than B and T cells and contain granules in their cytoplasm. Thus, NK cells are sometimes called large granular lymphocytes. Click the B cell. While B and T cells express specific receptors for antigen, no such receptors are found on NK cells. Yet, NK cells can recognize a variety of cells as abnormal, bind to them, and kill them. On the next page, we'll see how the natural killer cells recognize abnormal cells. Abnormal cells, such as cancerous cells, or those infected by viruses, often reduce the expression of certain membrane proteins. The suppressed proteins are those that tell the immune system that a given cell is self, in other words, that it belongs to the body. NK cells look for the absence of these self proteins. It's as if NK cells were saying, if I can't identify you as one of us, then you're a traitor, and I will kill you. Click the NK guard to see him identify the traitor.
Traitor!
NK cells and T cells are the two kinds of cells that continually scan our cells for abnormalities, a process called immune surveillance. They act in a complementary fashion. T cells look for the presence of abnormal antigens on the cell surface, while NK cells look for the absence of normally occurring self-proteins. How do NK cells kill abnormal cells? The same way that cells called cytotoxic T cells kill, a process we will explore in detail in Topic 6. For now, let's just say that killing involves direct contact and induces the target cell to undergo apoptosis, which is programmed cell death or cellular suicide. Click the traitor. NK cells are important in the early response to pathogens, acting to contain pathogens before the adaptive immune responses can take over. They continue to play a role after B and T cells are activated. Like macrophages, NK cells become more effective killers following activation by cytokines from certain T cells. Likewise, coating cells with antibody makes them better targets for killing by NK cells, just as coating pathogens with antibodies make them better targets for phagocytes. These processes show how innate and adaptive host defenses work together to protect us from infections. The first set of antimicrobial proteins we will consider are the interferons. Interferons are members of a larger group of chemicals called cytokines that modulate the immune system. Interferons interfere with viral replication, modulate inflammation, and activate immune cells. The three types of interferon, alpha, beta, and gamma, are distinct proteins but have common as well as unique functions. Gamma interferons act in a variety of ways to signal other immune and non-immune cells, which we will discuss further in Topic 6. Here, we will learn about the antiviral properties of alpha and beta interferons. Let's begin by examining how viruses replicate within cells. Click the virus to begin. Recall that viruses must enter cells to replicate. This is because a virus is little more than a protein-covered packet of nucleic acids, the genetic instructions for how to create a new virus. When a virus penetrates the target cell's membrane, the virus releases its nucleic acid and takes over the host cell's machinery to make more copies of that virus. Drag the virus into the left cell to see how it makes copies of itself. The presence of a virus replicating inside it causes the cell to produce and secrete interferons. Click the nucleus of the infected cell to see it produce and secrete interferons. Interferons bind to plasma membrane receptors on nearby cells. They act as a warning signal for as-yet uninfected cells, telling them that there is a virus on the loose. In response, the uninfected cells produce proteins that inhibit viral replication. These proteins act by degrading viral RNA and by preventing the synthesis of viral proteins. Drag the interferon to its receptor to see how it acts. Now drag the virus into the cell alerted by interferon to see if the virus can still make copies of itself. Chewing up viral RNA and blocking protein synthesis are not specific in that they work against any virus. In the short term, these mechanisms protect uninfected cells not only against the virus that invaded its neighbor, but also against any other viruses that may be in the area. The next set of antimicrobial proteins we will consider is the complement system. Complement gets its name from the fact that it complements or enhances other components of both innate and adaptive defenses. Like the blood clotting cascade, complement is actually a complex cascade of interdependent plasma proteins. As each protein is activated, it becomes an enzyme that activates the next protein until the final product is formed. When activated, these proteins can mark cells for phagocytosis, promote inflammation, and even kill some bacteria all by themselves. Drag the complement proteins to this bacterium to see it being lysed by the end products of the complement cascade. Let's examine the sequence of events in complement activation. This sequence might seem unnecessarily complex, but because of complement's potentially lethal effects to the body's own cells, its activation must be tightly regulated. Each step of the sequence provides a point of regulation that keeps the process from engaging until it is needed. Both the adaptive and innate defense systems can activate this cascade, but in different ways. As is often the case in the immune system, the enemy can be identified either by finding chemical markers that should not normally be present on cells, or by not finding chemicals that should be present. Click the bacterium to continue. Antibodies are chemical markers of the adaptive immune system that should not normally be found on our own body cells. Therefore, antibodies act to mark the enemy for destruction. One way they bring about this destruction is by activating complement via the classical pathway, which was so named because it was discovered first. Click the antibody to continue. Certain polysaccharides, or sugars, are found only on the surface of bacteria and not on normal body cells. When special sugar-binding proteins in the extracellular fluid, called lectins, bind these sugars, they trigger the lectin pathway of complement activation. We've already seen another type of lectin in action, the membrane-bound mannose-binding protein that triggers phagocytosis. Click the lectin to continue. The absence of inhibitory proteins, normally found on the body's cells, can also result in spontaneous complement activation. This spontaneous activation is the beginning of the alternative pathway. Click the two boxes that represent activation pathways due to the presence of chemical markers not normally found on our own cells. That's correct. Now find the other one. Each of these three pathways requires other complement proteins, for example, C2, C4, factor B, and factor D. But they all eventually cause activation of a protein called C3. Click the C3 to activate it. When C3 is activated, it splits into two fragments called C3A and C3B. These fragments have different roles. C3A causes inflammation, a process that we will study in detail later. C3B acts as an opsinin. Remember that opsinins enhance phagocytosis. Many individual C3s are split by the enzymatic cascade into many copies of C3A and C3B. Click the C3 to split another copy. C3b participates in cleaving another complement molecule called C5 into two parts, C5a and C5b. Click the C5 to cleave and activate it. C5a, like C3a, promotes inflammation. However, C5b combines with several other complement proteins on the surface of a bacterial cell to form a large pore called the membrane attack complex. Click the C5b to see the formation of a membrane attack complex. The membrane attack complex punctures the membrane, allowing water to flow in and solutes to flow out of a bacterial cell. As a result, the targeted cell lyses. Complement's ability to lyse some bacteria directly makes it a powerful defense mechanism. Even more powerful, however, are its other two roles. The ability of complement to opsinize a wide variety of pathogens, and its ability to enhance inflammation. Enhancing inflammation alerts and attracts elements of both the innate and adaptive immune systems. We will explore inflammation next. When the body is injured, such as with a cut, abrasion, or bruise, a sequence of events called inflammation is initiated. When the walls of the castle are breached, defenders and construction crews are called into the area. Similarly, in our bodies, cells and proteins are recruited to the area of injury so that defenses are ready and repairs can begin. We all experience inflammation many times throughout our lives. All of the itises you've ever had are inflammations. Usually these are short-lived problems called acute inflammations, such as tonsillitis, tendinitis, and laryngitis. But sometimes inflammation becomes a long-term or chronic problem, as in arthritis. In all of its various forms, inflammation is one of the most common problems that healthcare professionals face. Click the castle to continue. There are 4 cardinal signs or key characteristics that healthcare professionals use to identify inflammation. See if you can pick them out from this list. Drag your choices to the box. This sign might occur, but it is not a cardinal sign of inflammation. This sign might occur, but it is not a cardinal sign of inflammation. This sign might occur, but it is not a cardinal sign of inflammation. This sign might occur, but it is not a cardinal sign of inflammation. This sign might occur, but it is not a cardinal. This sign might occur, but it is not a cardinal sign. This sign might occur, but it is not a cardinal sign of Well done. The purpose of inflammation is to bring white blood cells and plasma proteins into an injured area. This action accomplishes 3 things. It prevents the spread of injurious agents, it disposes of pathogens and dead cells, and it sets the stage for repair. Click the injured finger. Chemical messengers called inflammatory mediators are central to this process. Virtually all inflammatory mediators cause 2 key effects. These are vasodilation and an increase in vascular permeability. Click the splinter to release inflammatory mediators. Vasodilation, or increased arteriolar diameter, leads to increased localized blood flow to the injured area. This increase causes both the redness and heat that are cardinal signs of inflammation. Increased vascular permeability occurs as the gaps between adjacent endothelial cells enlarge. This increased permeability occurs in capillaries and post-capillary venules, allowing plasma proteins and phagocytes to enter the tissues. Under normal conditions, white blood cells pass through capillaries and post-capillary venules without stopping. Inflammation requires that these cells exit the blood and enter the injured area. How does this come about? When tissues are injured, macrophages and other tissue cells release a variety of inflammatory mediators. In response to these messages, endothelial cells are activated, sprouting special surface markers called cell adhesion molecules. Neutrophils and monocytes bind to these molecules and stick to the blood vessel wall, a process called margination. Click the endothelial cell to see margination. As a result of binding to cell adhesion molecules in the presence of inflammatory mediators, neutrophils are activated and begin to make their way out of the blood vessel. The process of leaving the capillary is called diapedesis. Click the neutrophil to see diapedesis. Once in the tissue, neutrophils follow the trail of inflammatory mediators up their concentration gradient like a dog sniffing a trail, a process called chemotaxis. This process is how they find their way to the site of injury. Click the neutrophil to see chemotaxis. As part of endothelial cell activation, the clefts between endothelial cells become larger and leakier. This increased leakiness allows plasma proteins and more fluid than usual to leak into the injured area, causing edema, an increase in interstitial fluid. Edema causes swelling, which can also contribute to the sensation of pain. Click the capillary. Three important classes of proteins that enter injured tissue are antibodies, complement, and clotting factors. Tissue injury that triggers inflammation often introduces, and in some cases is caused by, the invasion of pathogens. If your body has already been exposed to these pathogens, then you usually have antibodies against them. In that case, your antibodies will bind to the pathogens and act as opsinins. Click an antibody to opsinize a bacterium. Click the capillary to leak complement into the interstitial fluid. When we discussed the complement system earlier, we focused on its actions as an opsinin and on its ability to form the membrane attack complex. In addition, two products of the complement cascade, C5a and C3a, are important inflammatory mediators. In addition to other actions, they increase vascular permeability and act as chemotaxins, that is, they attract more phagocytes to the area. Click a complement molecule to see complement act as opsinins and chemotaxins. Clotting factors also enter the interstitial fluid from the plasma and are activated in the tissues, forming a mesh-like network of fibrin protein that acts as a temporary scaffold for repair. It also holds the pathogens within a net-like trap, keeping them from escaping the injured area. Click the capillary to form a clot. This mechanism is such a powerful way of containing infection that some bacteria, such as Streptococcus, have evolved a counter-strategy, enzymes that break down the clot, allowing them to escape the net. Inflammatory mediators come from many sources. They promote vasodilation, increase vascular permeability, and act as chemotaxins. So far, we have mentioned that components of the complement system are inflammatory mediators, but other chemicals you are familiar with are inflammatory mediators as well. One of the most powerful inflammatory mediators is histamine, which is released by basophils and mast cells. While basophils are found in the blood, mast cells are found in connective tissue where they can react immediately to injury. Click the mast cell to release histamine. Prostaglandins and kinins are inflammatory mediators that are formed when cells are injured. In addition to enhancing vasodilation and vascular permeability, they activate pain receptors. Cytokines are principally involved in regulating leukocyte behavior and function, but they can also act as inflammatory mediators. Unlike the localized warmth of inflammation, fever is a generalized increase in body temperature. Various bacterial components, as well as cytokines released by macrophages, act as pyrogens. Pyro means fire, and gen is to create, so these are literally fire creators. They cause the body's thermostat, located in the hypothalamus, to set its temperature higher. Click the thermometer. It was once common practice to treat any fever with fever-reducing drugs such as aspirin, but we now realize that fever is a useful part of the body's defense system. Most pathogens do not grow as well at higher temperatures. During a fever, the liver and spleen sequester iron and zinc, making these essential elements less available to bacteria. Also, higher temperatures enhance phagocytosis and a variety of enzymatic processes used by the immune system. Unless the fever is high, it may be better to let nature take its course. Here's a summary of what we've covered. The first line of defense consists of the skin and mucous membranes and their secretions. The second line of defense, the innate internal defenses, consists of phagocytes, antimicrobial proteins, such as complement and interferons, natural killer cells, inflammation, and fever. Inflammation is a non-specific local response to injury that brings leukocytes and plasma proteins to the site of injury. Innate defense mechanisms may be insufficient to prevent or clear infections. Innate and adaptive defenses interact in many ways. Innate defenses often trigger and define the nature of adaptive defense responses. To test your knowledge, click the quiz button to go to the self-quiz. To access cross-references for this topic in your Benjamin Cummings textbook, click here. To test your knowledge, click the quiz button to go to the self-quiz.